Hi class, this is a review for Chapter 14, DNA, Genetic Material, and Chapter 15, Genes and How They Work. So let's go ahead and get started with the structure of DNA. I know off the top of your head you'd probably remember a few things like that DNA is double-stranded, that it contains genetic information, and all of that is true. So we'll go over that and also the chemistry part of the DNA structure. So it does have a 5-carbon sugar, which we call deoxyribose, which is right here on the bottom. This sugar, that's the deoxyribose sugar. It has a phosphate group, and it also has a nitrogenous base. So there are 4 different nitrogenous bases, and they're right here actually. A, T, G, and C. So adenine, thymine, guanine, and cytosine. Uracil will only be for RNA. But for DNA and in eukaryotic cells, although eukaryotic cells can have RNA, when we're specifically talking about DNA, the 4 nitrogenous bases are A, T, G, and C. If it is RNA, then it is A with U and G with C. And I'll show you a few more diagrams on that. You also have the hydroxyl group attached to the carbon sugar, which is right here. So the nitrogenous bases that we just discussed, adenine, thymine, guanine, cytosine, we know that they equal each other, or like that they pair with each other because of this rule that was discovered by Chargaff. And so Chargaff is the one that said that A equals T and G equals C, and there is actually like a mathematical weight to it. Like if you were to find the percentage of A, like example down here on the bottom, that A is 30%, then you would already know that T is also 30% because A equals T. So if you have one, then you can find out the other. Same with G. If G is 20, then C must also be 20. So that's kind of how that works. And if you were to add all 4 of these together, it should be 100, right? 30 plus 30 is 60, and then 20 plus 20 is 40. So 60 plus 40 is 100. Another thing that I wanted to go back to, actually I probably should show you that, is that when we say that A pairs with T, adenine with thymine, you must always have it that way. You cannot have A pairing with cytosine or A with guanine. It is only A with T and G with C. You can only have a purine pair with a pyrimidine. Purines have a double ring and pyrimidines have a singular ring. So you cannot have 2 purines put together, or on the opposite side of the spectrum, 2 pyrimidines. You can't have that. And so in this slide, you see how I have the purines, the 2 rings? Purines have 2 rings and pyrimidines have 1 single ring. And so that's how they're balanced that way, and that's how, I'm going to skip ahead really quick and then I'll come back. That's how the double helix has that structure, because of how slightly unequal it is, that you have a purine paired with a pyrimidine, and that's how you have the helical shape, and that's why, because of the purines and the pyrimidines. And I think that's all that I needed to talk about in this slide. As far as how we know that DNA is helical, is largely because of this woman right here. Her name is Rosalind Franklin. She doesn't probably get as much credit as she deserved, but she did discover that DNA was helical because of some experiments that she was able to conduct. So she did not discover that DNA is a double helix, but she did discover that DNA is helical. And then with that finding, these 2 men, Mr. Watson and Mr. Crick, what they did is they took the findings of Chargaff, that A equals T and G equals C, and then they took Rosalind Franklin's work, that DNA is helical, and then putting those 2 things together, they are the ones that came up with DNA being a double helix structure. And then also it says to note that they did not perform a single experiment related to DNA. All they did was they took the work of those other people and put it together. But these are the two people that kind of get all the credit for discovering that DNA is a double helix. This is the picture that we saw a little while ago. It kind of shows you two, I guess more of like the realistic way down here, and then more of the textbook, I guess, breakdown of how you would see it. But just know that DNA is helical. It is a double helix. It has two strands. It has these nitrogenous bases right here in the middle, and they are paired with a hydrogen bond that is in the middle. So right here between this G and C, there is a hydrogen bond there. Between the A and the T, hydrogen bonds. And it actually depends, and let's see if I had it there in another slide. I don't think we've gotten to it yet, but it does, it's here in the next one. Let's go right here. It does depend on, see, like G with C, it has 3 hydrogen bonds. Here with the adenine and thymine, it has 2 hydrogen bonds. But so what you're seeing right here is the same thing as this. So right here in the middle, these hydrogen bonds is what's holding these two strands together. And we say that they are anti-parallel because mostly this part right here, the 5' and the 3', they're not the same. They're not 5 and 5 or 3 and 3. It's 5 and 3, and so that's why they call it anti-parallel. So when we pair these nitrogenous bases together, we call that base pairing, that you're pairing the G with the C or the A with the T. That's called base pairing. I think maybe you might remember some people have like little sayings that help them. Like for A and T, it's audible tree, and G and C is gato cat. So if that helps you, try and jot that down so that you can keep it in your head. When DNA is being replicated, the way that it's replicated, we refer to that as semi-conservative. There were 2 other types that were proposed, but they were not found to be conclusive. So DNA replication does occur in a semi-conservative way, meaning that the daughter strands consist of 1 parent strand and 1 new strand. For this process to occur, you need something to copy, which is the parental DNA. You need enzymes to help, and then you also need the nucleotide triphosphates, which are building blocks. We're going to go over some of the really important enzymes when we're talking about DNA replication. This first one is DNA polymerase, and there's 3 different polymerases. But the first one is going to match the pairs. So if there is an A, DNA polymerase is going to go find a T nitrogenous base. The same if there is a G or guanine, it's going to go find a cytosine. So it actually brings the complementary nucleotides and puts them together. And then notice the direction that it's happening in. So it's going from 5' onward. One of the enzymes that helps unwind is this one called helicase. And so when it is unwinding the DNA, let me go back to that picture that we were looking at. What it's going to do is it's going to break these hydrogen bonds that we talked about earlier. And so that's how it's able to unwind DNA. It breaks these hydrogen bonds, and let's see. I'm trying to see if I have a picture for you. I don't think I have one yet, but just keep that in mind that helicase does unwind the DNA. And another picture will show up in a little bit. Replication is semi-discontinuous. And we say that it happens that way because there is what we call the leading strand and the lagging strand. And so for the lagging strand, it does it in pieces, and which we call those the Okazaki fragments, because Okazaki is the last name to the person that discovered it. And so that part is happening in pieces. And the other one is happening just continuously, where polymerase is there adding nucleotides in a uniform and just fast way, versus the other one, the lagging strand, it's happening in pieces. So you can see here's a fragment right here in blue, and then this should be another one also in blue. So what it will do is it'll bring in nucleotides in chunks. Instead of bringing a nucleotide singularly, it brings it in a whole chunk and just kind of pieces it together. And so that's how this one is different from the leading strand. And let's see. I know this looks like a lot is going on. And granted, there is a lot going on. Let's see if we can break this down the best that we can. So you have your original DNA, the parent DNA. And so helicase is right here at what we call the replication fork, and it's breaking these hydrogen bonds. And then, let's see, where is polymerase? Well, here's one polymerase. Where's another polymerase? I guess this is another one right here, but it doesn't have it labeled and we know that it is polymerase because it's adding bases. So this one is what we call the leading strand. And we know that it's a leading strand because it's adding nucleotides continuously one by one. Right, added them one by one, one. Versus over here, it's adding them in pieces. So here's a piece right here in blue. There's another piece. There's another piece. And so this is happening on going this direction, and this one is happening in this direction. That's why you have these arrows. So for the leading strand, it's going from 5' to 3', versus for the lagging strand, it's going from the 3' to 5'. So hopefully that helps a little bit. So again, you have DNA replication occurring. The way that it replicates is that these hydrogen bonds are going to be broken on one side of the DNA strand. This is still parental DNA. Oh, that's another thing that we should mention is that for the two strands of DNA, each of them do get one copy from the parent. And then they get their new strand, which is made complementary. But yeah, the hydrogen bonds are broken. This one is happening discontinuously in fragments, which we call the Okazaki fragments, and it's also called the lagging strand. On the opposite side, it's happening all seamlessly, and this is what we call the leading strand. So the next part, this is starting chapter 15, the genes and how they work. When we were talking about replication for DNA, this does happen in the nucleus. Just wanted to make sure that you remember that does happen in the nucleus. And then when we're doing the other parts, transcription, translation, not all of these happen inside of the nucleus, and we're going to decipher which one is which. So the way that this occurs is that it first starts with DNA, and then DNA is made into RNA, and then RNA is then made into a protein. So from DNA to RNA, we call this transcription. From RNA to protein, we call that part translation. Transcription does happen in the nucleus, and translation does happen in the cytoplasm right outside of the nucleus. So it kind of shows you a little bit slightly from DNA to RNA, and then that is translated to a protein. So when we're reading DNA, we read it in what we call like triplets, and the correct term for that is a codon. So codon is a triplet of nucleotides. So right here, here's one AUG, that is one codon. CCU, that's another codon. And then ACC, another codon. So, but if you can tell, some of these, it says that one has been deleted. So there's a mistake going on here, but let's see. Let's find you a better one. So I guess all of these have mistakes, but just, let me go to the chart. There we go. This is probably much better. So you see how these are in triplets? All of these are codons, and each codon corresponds with an amino acid. So here, there should be 20 amino acids on this page. So I don't want to count, but I'm pretty sure that there's 20. So just remember, codons are triplets of nucleotides and they code for a specific amino acid. Some of the things that could go wrong is a mutation. Mutations are not always bad. The mutation can bring genetic variation and it could be good. So when you see the word mutation, it doesn't necessarily mean that it's going to lead to a bad thing. Mutations happen by chance and they could be either good or bad or just neutral. It really does depend. But just know that mutations do occur. It could be when you add in something, you delete something. And then if you get, actually, let's probably emphasize this. Let's go to that codon chart. So notice how these codons code for a specific type of amino acid. So if you have a mutation, the amino acid that it was supposed to code for will be coded for something else. It'll be switched, right? So if you were supposed to have a code that had, I don't know, let's say these 3 amino acids, but then the code changes, well, then your protein is not going to have these 3 amino acids. It could have maybe these 3 amino acids. And so it's not going to code for the same protein. So if you have some change up in your DNA, the protein at the end that's going to be made is not going to be the same, which could be good, it could be bad, or it could be neutral. So just kind of keep that in mind. It's not always a bad thing to have a mutation. That could very well be a good thing. When your DNA is turned into RNA, there is a part that is not really, I don't think, talked widely about, but you have a lot of genetic material that is not needed, and we call that non-coding DNA or non-coding sequences that we call introns. And then there's also the part that is coding or that will be coded, and we call that an exon. So let's look at this picture on top, and it has it color coded for you. So the blues are the exons, the the purples are the introns. And then what's going to happen is that anything that is termed non-coding, which is an intron, is going to be spliced and removed. And then the only thing that is left is the exons. So notice here how it says the introns are removed. That's because they are non-coding. We don't need them. We remove them, and then all that's left is coding, coding sequences. And this just kind of tells you that there is a protein that helps kind of do that process. So that's interesting. And also, you don't really need to know this for your exam, but for prokaryotic cells, they have much less non-coding DNA, which is interesting. And maybe if you take a higher level, like a genetics class, they'll tell you why that is. In the translation process, tRNA helps a lot because tRNA will bring the anticodon, which will then help lead to create your protein at the end, and we'll show you a picture in a little bit. The ribosome is where also translation occurs. And here are my tRNAs. And then let's just kind of end it with this picture. So for transcription, again, the DNA is located in the nucleus. It then gets transcribed into RNA. After transcription, it's going to leave the nucleus. Now it's in the cytoplasm. And then you have the tRNAs that bring anticodons and kind of place them on there with the help of the ribosome. And then they start to grow what we call an amino acid chain. And then at the end of that process, you will have a protein, because all that proteins are is just a lot of amino acids put together. You might also see the term polypeptide chain. That's the same thing. So a polypeptide chain is a protein or will lead to a protein. So just kind of be familiar with those words. And that's really it. So thank you guys. I hope this helped somewhat, and then good luck on your test.